Hi, I'm James Peterson. I'm a professor in the chemistry department at the University of Pennsylvania, and this is my lab's Virtual Philly Materials Day presentation. So today, I'm going to tell you about the background of some of the work that we do, and then two of the graduate students in my lab, Chloe and Kristen, are going to tell you about the details of how some of the experiments are done.
Hi, I'm Chloe.
Hi, I'm Kristen.
So we're going to tell you about bright molecules for brain studies. And what that means is we're going to tell you about how we use the brightness of fluorescent molecules in order to study how certain proteins in the brain contribute to neurodegenerative diseases like Alzheimer's disease and Parkinson's disease. And the way we're going to do that is by doing experiments that are similar to the way that CGI movies are made. So on the left, you can see a guy in a suit with a bunch of little balls on it, and those balls are recorded by the camera. And so by recording how those move, they can then represent the fact that he's waving his hand and use that to make a model of him that can be used to make a CGI movie. In the same way, we're going to take proteins and attach fluorescent probes to them that allow us to watch how those probes are moving and therefore how the protein is moving, either by changing its shape or by moving around inside of neurons. Now, the types of protein shape changes that we're going to look at contribute to a number of neurodegenerative diseases, including Alzheimer's disease, in which you see memory loss, Parkinson's disease, in which you also see shaking and loss of control of muscles, ALS, also known as Lou Gehrig's disease, and chronic traumatic encephalopathy, or CTE, which you may have heard of a lot about recently in terms of injuries to head injuries to athletes. So all of these diseases have in common that they involve the misfolding of proteins. So proteins in the body folding up into the wrong shape. And so when that happens, it can cause them to aggregate and clump together and form these fibrils that you see forming there. And this is actually similar to something that you probably have seen all the time, which is the cooking of an egg. So when you have a properly folded protein in the egg, then you have a clear, almost liquid. But when you cook it, of course, then those proteins unfold and clump together, similarly to the way the fibrils form, and the egg white turns white. And so what we're trying to do is put those fluorescent probes onto proteins so we can track how they're changing their shape when they fold up on each other and form those fibrils. So they have to change shape and stack many copies of the proteins on top of each other to form those fibrils. And we want to see just how that process takes place. And so if that fibril clumping does happen, then they can start to accumulate inside neurons. And those clumps are things that you can see if you actually slice open a brain, and then you can see these dark clumps forming. And of course, those dark clumps are not good for the neurons. And so when they accumulate, that can lead to neuronal death. And so you can see a picture of a single neuron with a clump there in the bottom right. But what we're really worried about is spread of that neuron toxicity to other regions of the brain. And so after someone with Alzheimer's or Parkinson's disease dies, if you take sections of their brain, you can see these dark clumps of protein and sometimes even holes in their brain where neurons have died and left space. And so we want to be able to move beyond just trying to understand what happens to someone's brain after they've died, but to be able to actually image these things in live brain imaging, and so doing that both in models with neurons as well as in actual human brains. And so we're going to tell you a little bit about how we do that using fluorescent molecules.
All right, so first off, the way that we will kind of track this protein is by adding on a fluorescent molecule. And so the fluorescent molecule that we make in the lab is called acridonyl alanine, or acridone. We can make it from tyrosine, which is actually made in your body and pretty cool, in a few simple synthetic steps. The last step of which is right here, where we basically put in this molecule right here, and we boil it in what's called a redox condenser. And so it's heating, it's this trap right here makes sure that it's not evaporating. And at the end, we get out a mixture of products because we have what we want and what we don't, and it becomes kind of this big brown mess right here. So what we do next is we purify it on biotage, in which we have solvents running through to purify our sample that we want away from the stuff that we don't want. Problem with this is that it's pretty dilute, which means that there's a lot of extra water and other solvents that we don't want. And so what we do next is we put what's called a rotary evaporator, or rotovap. And this basically gets rid of all the extra solvents and makes it so that we have a nice concentrated sample. And so one of the ways that we can kind of check to see what's going on and make sure we have made acridone or blue fluorophore is by shining a black light on it. And as you can see, there's a nice, beautiful blue fluorescence.
So now that we've synthesized our blue label, now we want to incorporate it into a protein. And so to make this protein, we actually hijack bacteria and have it do all the work for us. And so if we have a plasmid or DNA that codes for our protein of interest, if we put these bacteria under stress, for example, if we heat them up, they'll take in things from their surroundings, so they can take in that plasmid. And then if you grow them on a nutrient-rich media, then they will actually grow into these colonies, as Chloe is showing you here. And so each one of those colonies or spots corresponds with a bacteria that took up our plasmid and then was able to grow. And so what she's doing is she's now setting up this primary culture. So she's taking a single one of those colonies, she's streaking it, as we'll call, and then putting it into this liquid media. So once the cells have grown to a certain point, they need more room. So then we'll transfer that primary culture into a larger culture. And as you can see here, we're putting it into our shaker. So the shaker keeps it aerated and keeps the cells happy and alive. And so we'll add in a specific chemical that then tells the protein to start tells the cells to start producing our protein, and then we'll purify it on an FPLC system. So this is actually in a deli fridge to keep it nice and cold, because these proteins prefer to be at colder temperatures. And so then we can purify out our protein with our blue label on it, as you can see in these fractions here.
So now that we have our labeled protein, we want to look and investigate the distances between all of these points and kind of map out what's going on with our protein. We do this with the technique called FRET. And what FRET basically means is that when two fluorescent molecules are close to each other, one fluorophore can donate its energy to the other one. So in this case, the green fluorophore will donate its energy to the red one. And we do this in an instrument called a fluorimeter, which is right up here. These are actually videos right down here of the fluorimeter, but it's in the dark, so you can't really see what's going on. So I have two samples that I'm measuring down here, and one of them is synuclein with fluorophores that are way far apart from each other, like so. So the idea is that this green fluorophore is way too far away from the red one to donate its energy. And so I'll put this into the machine like so and take a measurement. And what we can see is kind of almost like a fingerprint for the fluorophore. So this is a green fluorophore trace right over here. And then over here is where we should see red if they were close together, but in this case, they're not. And so we're not actually seeing any red fluorescence. Now, if the fluorophores are close together, then this green fluorophore has a chance to give its energy over to this red fluorophore. So if we look at this video right down here, again, I've put in the sample, I close the door, and I have the curve up that we had from before, this one over here. But now that the two fluorophores are close together, we're going to measure and see what happens. And so right away in this green region, we can see the decrease in the fluorescence intensity right here. This peak is smaller, and this red peak over here is larger, meaning now that they're closer together, the green fluorophore is just giving all of its energy over to the red fluorophore. And so what we can do is map out the protein structure by changing the positions of where we put these red and green fluorophores and measuring out all their distances. And that's how we get this kind of fibril model of what's going on inside of alpha-synuclein fibrils.
So we've shown you how we can synthesize our blue label, but actually if you change the chemical structure of that, you can get out all of these different colors. So you can get out green, red, yellow, etc. And so one place where you may think of all these different fluorescent molecules and colors would be with glow sticks. But something that you might not know is that for a glow stick alone, if you shine a UV light on it, you can actually see it glow. And so this is pretty transferable or comparable to what we do in our lab. So imagine that this UV light is actually this pretty fancy fluorescence microscope. So if you were looking onto a plate of cells with this instrument, you wouldn't really be able to distinguish anything. However, if you were to label different organelles, cell structures, or proteins with different colors, then you can be able to study and track what's going on in these cells. So in our lab, for example, we could label the protein synuclein. When it's clumped together, we can label it so that it's green. And then you can watch as that protein travels from a diseased neuron to a healthy neuron, and therefore kind of understand how the disease could propagate. And so down here, this is actually a real image of neurons where those clumps of that protein is labeled green, watching as they move.
Thanks for watching, and I hope you found what you heard interesting.
So besides myself and Chloe, there are a lot of other members in our group who do all of these different experiments. And this is back in 2019 when we could gather together for a photo.
If you're interested and want to hear more information, please check out our website down here at the bottom or follow us on Twitter. Thank you.
Bye.